We are going to cover chapter 26 on population genetics. So by the end of this chapter, you should be able to define and understand what a natural population is. So this can be a human population, or it can be a population of other natural wild organisms. And what you want to be able to do is analyze the genetic differences that are segregating within that population. And so two of the really important ways you measure genetic variation is using allele frequency and genotype frequency. So you're going to want to understand what those mean and how to calculate them. No Winn-Hardy-Weinberg equilibrium applies. So this is a way to connect allele frequency to genotype frequency, and a way to calculate those two numbers if you're given one. And so this only applies under certain conditions. So make sure you understand when those conditions apply, and then if they do, how you calculate the allele frequency from genotype frequency or vice versa. And then finally, understand how evolution can lead to changes in allele frequency over time. And so those processes, those evolutionary forces, can be genetic drift, they can be natural selection, they can be bottlenecking, and they can be migration. And those all have very profound effects on allele frequency and also genotype frequency. So understand kind of what causes them and what they mean. And then finally, how linkage allows us to identify close relationships. So typically, genetic variants, polymorphisms, are unconnected to each other in natural populations unless you're closely related to an individual. And so that gets back to your chapter 5 knowledge of recombination and how that will link nearby genetic variants on the same chromosome and how we use that to identify close relationships. And these are kind of the types of analyses that allow us to do Ancestry.com or 23andMe type approaches to predict kind of your historic ancestry. And then this linkage is what we use in order to identify close relationships. And so these light purple and dark purple bars are runs where your genetic variation is identical to another individual. So for my brother, you can see that on chromosome 1, the parts of the chromosome that are identical to each other. And as you go further and further out into more distantly related individuals, like a second to third cousin, the amount of those runs gets smaller and smaller. But this statistical significance, because it carries so many different variants, is very high, and so you can very reliably determine that this individual must be a close relationship to me. And so kind of the most basic part of this chapter is just understanding what a polymorphism is. So we are not identical to the human reference genome, and on average, 1 out of every 1,000 nucleotides is different. And so that indicates that, we are very similar to the reference genome. 99.9% of our base pairs will be identical, but because the genome is so large, that indicates that there's a very large number of actual differences between us and the reference. So, a good ballpark number to have in your head is there's about 3 million differences between you and the reference. And so there's a lot of actual individual differences. And so these can be things like single nucleotide polymorphisms, where we just have a change of one of the nucleotides to one of the other 4 nucleobases. So here we've highlighted an example where, let's say, the reference was the C in this position, but actually your maternal chromosome has a T in this position as well. So this is an example of a single nucleotide polymorphism. Individual 2 matches the reference at both locations. So one of the things that's important to remember is that the differences in you are not likely or not necessarily the same as the differences in another individual. But there's also things like short tandem repeat polymorphisms, so CAG, 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 and so due to kind of genetic slippage during DNA replication, you can actually increase or decrease the number of these repeats. So this is a different type of polymorphism. And there's other ones as well, such as insertion deletion polymorphisms and also larger rearrangements of DNA. The large majority of differences fall within this single nucleotide polymorphism type range. So the other thing to remember about these polymorphisms is that they can have either very profound effects on the phenotype or they can have no effects on the phenotype, and that just determines where they fall within the genome. So this location right here, this could be in a coding region, and so this would change the amino acid of the encoded protein. It could fall in the promoter region, and so this could affect the expression levels of the gene that it's connected to. Or it could just fall in a part of the DNA that has no effect on expression or protein function. And in that case, it would have no effect on the phenotype of the organism that carries this polymorphism. And so this was actually a very profound discovery or very profound theoretical consideration that was made, is that the large majority of polymorphisms that are within you, these 3 million differences, we now think that the majority of those don't have any effect on the phenotype. So these are very useful from a sense of understanding ancestry, but the effect on why you are you, the majority of these probably have no effect on any phenotypic differences between you and other people. And typically when you compare individuals that are completely unrelated to each other, in general, if you look at the polymorphisms, they are completely unrelated. So if you have an A in this position and a C in this position, in general, if you look at other individuals, in general, these two polymorphisms are what we would say are completely unlinked to each other. And so that means it's almost like rolling a dice 2 times, where the effect of rolling the first dice has no effect of rolling the second dice. And so this is kind of how you should think of polymorphisms in unrelated individuals, is each one is kind of, if it's an A or a G, whether or not this is an A or a G has no effect on whether this next one is a C or a T. All right, so we are going to cover this in 4 different parts. So we're going to talk first about populations. What is the definition of a population? And then how do you measure genetic variation within that population? Then we're going to talk about Hardy-Weinberg, and then part 3 will be the evolutionary forces that change allele frequencies, and finally part 4 will be using linkage to infer ancestry.